Okay, so I will hand it over to Marie Marsh so she can talk about the energetic importance of the Gulf Coast Barrier Island for the spring stopover ecology of long-distance migratory birds.
All right. Good morning, everyone. I want to thank you all for being here on the Friday prior to spring break here in Bell State, and I appreciate your guys' patience. I know the flyer said it was 10 o'clock. That was a misprint. We were supposed to start at 10:30. We started a little later due to technical difficulties, but everything's all right. And now I'm glad to talk to you guys about my project on Barrier Islands, Florida. As Dr. Heckscher mentioned, I'll be talking about spring migration. Been fully immersed in avian migration and spring stopover ecology research for the last 3 to 4 years. And it's been my pleasure working for Dr. Heckscher and working within the NOAA ECSC. I've had a ton of fun working along the Florida Gulf Coast, working here in Delaware at White Creek State Park. And interesting aspects of my work. So let's get started. Nearctic neotropical migratory birds are species that nest in the United States and Canada and migrate south to overwinter in Mexico, Central and South America, and the Caribbean. This animated map was created from eBird citizen science observations of 118 species compiled over 1 year. This is a great illustration of the remarkable migration these species undertake twice a year that encompass nearly the entire hemisphere. Migratory birds are critical for biodiversity. They provide important ecosystem services, and they represent an important internationally shared resource of conservation concern. And this last part is incredibly important because many of the populations of migratory birds have experienced precipitous declines in the last 4 decades. And these declines have been attributed mainly to anthropogenic causes: habitat alteration, habitat modification, fragmentation, introduction of non-native species. The list goes on and on. During migration, survival rates are very low. Individuals, when they're migrating, have to contend with avoiding predators, finding sufficient resources, avoiding bad weather, and confronting novel pathogens. And they do all of these things usually in unfamiliar environments. So it really is remarkable. So how do birds make these long distance migrations twice a year? First, birds use fat to fuel migration. Most songbirds embark on long distance migratory migrations punctuated by short stopovers where they rest and rebuild these energy reserves. During this period of migration, in-route habitat is fundamental for the successful completion of these migrations. Stopover habitats are the sets of habitats that migratory birds use during spring and fall and are important because they provide places to rest, refuel, and avoid predators. The frequency a bird will make a stopover and the duration that the bird will spend at a stopover site is influenced by prevailing weather, the individual's body condition, their assessment of the risk of mortality, either by predators or exposure to weather, and the resources available at these sites. However, not all stopover sites are created equal. To classify stopover sites, we use the conservation framework where we have a continuum of habitats that fall into 3 major categories. First, we have a full-service hotel. And here we would see an extensive area predominantly forested where the available resources, the necessary resources for migration are relatively abundant and available. At a convenience store, a bird can briefly stop and replenish some of their fat or muscle. But at a fire escape, the resources available are too low for a bird to really replenish fat stores or muscles. But just as in a building, a fire escape allows survival, and it allows birds to continue the migration after staying at a fire escape stopover site. I'm particularly interested in these types of stopover sites, the fire escapes. They're the habitats that migratory birds would encounter after crossing significant ecological barriers, such as the Gulf of Mexico, or where they're compelled to use because of emergency situations. The barrier islands along the Gulf Coast are prime examples of fire escape stopover sites. Barrier islands are important because they provide ecological functions and they protect our coastlines from severe storm damage. Barrier islands are also very important for wildlife. For migratory birds, they provide the first refueling opportunity after crossing the Gulf of Mexico, and they can be especially important for fat-depleted birds. While barrier islands are important features along coastlines, they're also vulnerable to sea level rise. Here we see East Ship Island in Mississippi before and after Hurricane Katrina. The storm severely widened the breach between East and West Ship Island. And here I also want to do a quick plug. Dr. Heckscher's lab just came out with sea level rise barrier island effect on migratory birds that was just published in PLOS ONE. So check it out, it's pretty good. So migratory birds have been known to use these barrier islands during spring and fall migrations. These are data of arrival fat, which is an indicator of body condition, for 2 islands in the western Gulf Coast of Mexico. In fact, one of the islands in this study was East Ship Island, which is the island we saw in the previous slide. On the y-axis, we have % of birds captured, and on the x-axis, we have 6 years of study. The majority of the birds that arrived on these barrier islands arrived with little to no fat reserves. And again, as I mentioned, the Gulf of Mexico is the largest barrier, ecological barrier migrants encounter along the route. However, successful migration translates not only into individual survival, but also into timely arrival to your territory. And this is especially important in the spring, when earlier arrival allows males to establish and defend territories and find their mates. For example, in American redstart, earlier arrival influenced the reproductive success in males. Here we see that males that are successfully mated, these guys, arrived significantly earlier than the birds that did not. This illustrates the selective pressure of minimizing your migration time. So birds might be pushing the physiological limits in order to be the first ones on site. But pushing your physiological limits or not encountering adequate stopovers along your route may have important carryover effects. Here we see the relationship between female fat content upon arrival on the breeding grounds for American redstarts and the relationship with the average egg mass per clutch. Females that arrived with higher fat scores had increased reproductive success compared to females that arrived with lower fat scores. And many studies have found that young birds have less experience foraging and avoiding predators and are generally outcompeted by older birds. In this study that actually used geolocator data, they found that adult wood thrush migrate at a faster speed and stop over less frequently than young birds. In migrating birds, body mass change during stopover is used usually to evaluate the quality of the stopover site. So traditionally, what we would do is we would catch a bird and weigh it and let it go, and then on subsequent recaptures, we would look at whether it gained or lost mass. On the left, we see that longer stopover, so we have body mass and duration of stopover. And on this graph, we see that longer stopover resulted in increases in mass. And this is in the Beery at a stopover site in Colombia in the fall. On this other graph, we have the rose-breasted grosbeak in spring on East Ship Island. And longer stopovers actually resulted in decreases in mass. Here, this method is used to compare 2 stopover sites to the wide-eyed vireo. This is, again, East Ship Island on on the top graph, where stopover duration really did not influence mass gain. But at Chavito Woods, which is a relic Chenier coastal forest in Louisiana, longer stopovers resulted in increases in mass. So this is a really cool way to compare two different stopover sites. However, there's a disadvantage to this method. You have to rely on repeated measures of recaptured individuals. And due to the transient nature of migration, recapturing an individual is really difficult. You end up with really small sample sizes. So because of that, there's been a bunch of other different kinds of methods developed to try to get this kind of data from larger sample sizes. And that's the first tracks by time of day method that uses the body mass measurements of all captured individuals and is regressed by time of day. And the idea is that a significant positive relationship would indicate that birds that were captured early in the morning were lighter than birds captured later in the day, and would imply that the birds were gaining mass at the site. However, there's some drawbacks with this method in that it needs to have a large sample size, and we're assuming that all birds in the sample were at the stopover site for the same amount of time. So the really cool new method to avoid all of these drawbacks is the use of plasma metabolite profiling. And plasma metabolite profiling offers a way to assess refueling performance of migratory birds from a single capture. Birds use glucose and glycogen for muscular activity, but as I mentioned earlier, they rely on the accumulation of lipids to fuel long-distance migrations. And various lab and field validations have confirmed the use of plasma metabolites, especially triglycerides, as an estimate or to a qualitative measure of refueling rate. So in this graph, this graph indicates a positive relationship between increasing mass and higher levels of triglyceride concentrations. In birds, triglyceride concentration rapidly increases with the intake of food, so elevated plasma triglycerides are indicative of fat deposition. And this method has been widely used now for, 6 or 7 years. And for example, in this study, they looked at plasma triglycerides as a tool for assessing refueling rate. And they compared the triglyceride concentrations from high and low elevation stopover sites for the fox sparrow in the fall. And they found that they refueled at a greater rate at higher elevation, this one right here, at higher elevation stopover sites. This graph also indicates that refueling rate increases throughout the day for both sites. Plasma metabolite profiling has also been used to detect differences in refueling rates of different groups. At an urban stopover site in the spring, they found that male yellow-rumped warblers refuel and male common yellowthroats refuel at higher rates than females. So now for my study, the overall objective of my master's thesis was to assess the refueling performance of migratory birds using the Apalachicola Bay Barrier Islands during spring stopover. I aim to confirm the use of the island by spring migrants, to determine its energetic importance, and to provide data to inform conservation and management decisions. I tested the hypotheses that longer distance migrants would arrive in poorer condition than shorter distance migrants, and that because of this, longer distance migrants would have lower refueling rates than the shorter distance migrants. And I would also test it to see if there were any age or sex related differences. My other hypothesis was that subadult birds would arrive later in poorer condition and refuel at lower rates than adult birds, and that males would arrive earlier in poorer condition and refuel at higher rates than females. However, due to technical difficulties, this last hypothesis was not tested. My study site was located on beautiful St. George Island, Florida, within the Apalachicola Bay. The island is 20 miles long and averages less than a mile wide, and it sits about 4 miles from the mainland. On the Gulf side of the island is a narrow band of beaches and low-lying sand dunes that grade into mixed woodland grasses, palmetto, and bayside marshes. My research was focused on 4 species of migratory birds: the gray-cheeked thrush, the veery, the Swainson's thrush, and the gray catbird. I selected the Gutierrez thrush species because most individuals that breed in eastern North America make transgulf migrations in the spring. The gray catbird is not a thrush. It's a mimid, but it is of comparable size and mass to the thrushes, and the species is relatively abundant. It's relatively common on the island during spring. While the Gutierrez thrushes are insectivorous, the catbird has a more generalist diet, and all 4 species are sexually monomorphic. Gutierrez thrushes are long-distance migrants that breed in the northeast in boreal forest and overwinter in Central and South America. As we know from research conducted right here at Delaware State by Dr. Heckscher, we know that the veery conducts one of the longest distance migrations of a neotropical migratory bird. The gray catbird is a shorter distance migrant. It breeds along the East Coast and overwinters in southern United States into Mexico and Central America and the Caribbean. In comparison, the gray catbird has a wider distribution during the wintering time and a wider breeding range compared to Gutierrez thrushes. My study site, again, was located on St. George Island within the Apalachicola National Estuary Research Reserve boundaries. The Heckscher Lab established a constant effort mist netting station that operated daily during peak migration in 2013 and 2014. Nets were operated daily and checked every 8 to 10 minutes, and every bird captured was banded, aged and sexed, measured and weighed, and safely released after all data were collected. To determine body condition, I used standardized methods to assess pectoral muscle score and to determine fat content. And as I mentioned earlier, fat is the fuel of long-distance migration. I collected blood samples via brachiovenipuncture using a 26-gauge needle, taking a maximum of 1% blood volume. All the samples were held on ice for 4 to 6 hours while we were in the field until I was able to take them back in the evening and spin them in the centrifuge, separate the plasma, and put it on ice, on dry ice or liquid nitrogen until laboratory analyses. Plasma triglyceride concentration varied quickly due to changes in feeding state, so I kept track of capture and bleed times. Now back in the lab, plasma samples were assayed using a commercially available triglyceride kit. I increased the volume of all the samples 3-fold using saline, ran the samples in duplicate in a 96-well microplate spectrophotometer, and measured the absorbance at 540 nanometers. For molecular sex determination, remember all these species were sexually monomorphic. I extracted DNA from residual red blood cells and used PCR to amplify the sex chromosome-based CHD gene, and using the P2P8 primers, and then visualize it in a 2% agarose gel. And the idea was that females will present 2 bands and males would present 1 band. You can see that down here. As I mentioned earlier, I had some technical difficulties with this technique, and due to time constraints, this part of my research was put on hold. For statistical analyses, I checked all the variables for normality and homoscedasticity. I interpreted results as significant when P was less than or equal to 0.05, and I ran all of the stats in R. Okay, so what did I find? We have a similar trapping effort both years, and that's net hours. But trapping effort resulted in low numbers for the thrush species. So after ensuring that there were no significant differences in a number of variables, I decided to lump all the thrushes together to facilitate analyses. And this gave me 2 groups. The long-distance migrants, the thrushes that I'm calling long-distance migrants, and the gray catbirds that are the short-distance migrants. For simplicity, I'm going to refer to these as thrushes and catbirds. And I ended up with a sample size of 63 thrushes and 186 catbirds. I wanted to see if there were any differences in the age structure of birds using St. George Island. So I ran a chi-square task and found no significant difference between the age structure of thrushes and catbirds the first year, and no significant difference in the age structure between thrushes and catbirds the second year. However, for the gray catbird, the proportion of age varied between years. The first year I had a higher proportion of subadult birds, and the second year there was a significantly higher proportion of adult birds the second year. Subadults and adults. I'm sorry, the terminology can be kind of. Next I examined the differences in arrival dates. I ran a Mann-Whitney U-test, and I found a year effect. First, there were interannual differences. Thrushes in 2013 arrived later than the thrushes in 2014. And here we have Julian date and my two groups and years. And I also found significant difference for catbird arrival between years. There was an intra-annual difference. But also, I found a migration strategy effect. In 2013, gray catbirds arrived earlier than the thrushes. And in 2014, again, catbirds were arriving earlier than the thrushes. Next, I wanted to look at capture times, the time they were arriving on the island. I used, again, a Mann-Whitney U-test, and I found no significant difference in the capture time for thrushes between years, and no significant difference in the catbirds between years. However, in 2014, catbirds arrived earlier than the thrushes, earlier in the day. So now I wanted to look at body condition upon arrival. The first variable is fat score. Remember, fat is the fuel of migration. I found no significant differences in fat reserves for thrushes between years, so it was similar fat content in 2013 and 2014 for thrushes, and the same for catbirds. There was no difference between years. However, thrushes consistently arrived with lower fat reserves than catbirds. I'm not presenting this here, but I found no age effects in fat reserves on arrival for either thrushes or catbirds. The next variable that I looked at was pectoral muscle score. For thrushes, there was no significant difference between years in their arrival muscle score, but for catbirds, there was a significant difference. Thrushes arrived, catbirds arrived with higher muscle scores in 2014. The more interesting result, though, is the difference between thrushes and catbirds. Thrushes consistently arrived with lower pectoral muscle scores than catbirds. And again, I'm not showing it here, but I found no age effects in pectoral muscle score on arrival for either thrushes or catbirds. The next final indicator of body condition was size-corrected body mass. And here we're looking at the size-corrected body mass of the gray catbird for both years. And I ran a t-test, and this is textbook. There's no difference, no significant difference in size-corrected body mass for catbird. A linear regression between size-corrected body mass and Julian date revealed that there was no significant difference in the first year. But in 2014, I've had a significant relationship between size-corrected body mass and Julian date. This means that lighter birds were arriving earlier in the season. I ran another linear regression between size-corrected body mass, but this time against time of day. And I found no relationship in either year. This means that the body mass for gray catbird was not increasing throughout the day. Now looking at thrushes. I have the size-corrected body mass of thrushes and the 2 years of study, and here we have a significant difference. Thrushes were significantly heavier in 2014. Recall that we just saw that catbird had no significant difference in size-corrected body mass. For brevity, I'm not going to show you all the graphs, but I did the same regressions for the thrush data, and I found no significant relationship in Julian date or in capture time. All right, now the really interesting stuff. I used plasma triglyceride concentration as an estimate of refueling rate, and I found interannual differences. So thrushes, so let's just look at thrushes, and these are thrushes, the first year and the second year, and there was a statistically significant difference in the triglyceride concentration between the two groups. Now looking at catbirds, we have the same pattern. There's a statistically significant difference. However, intra-annual refueling rates of thrushes and catbirds were similar. And the ones in the box are the triglyceride concentrations for 2013, and there's no significant difference there. And these are the concentrations for 2014, and there's no significant difference there. For brevity, I'm just showing gray catbird data, but I found that there was no age effect on refueling rate for these species. Here we have second-year birds are subadult birds, and after second-year birds are adult birds, and there's no significant statistical difference between the triglyceride concentration between the two groups, and the same for the two groups in 2014. And I found a similar pattern for thrushes. So I wanted to look at the refueling rate a little bit more closely, and because of these, there was no differences, I lumped all of the thrushes and catbirds together just to look at it a little bit more. And I found that plasma triglyceride concentrations were higher the first year, 2013, than they were in the second year. There were significant differences. And I ran a linear regression, and I found that refueling rates did not increase throughout the day for either study year. Finally, I aimed to identify the variables that contributed to differences in triglyceride concentration based on migratory strategy and study year. So I ran a generalized linear model with a backward elimination procedure using the step command in R to drop terms until I found the best fit model based on AIC values. And these are the best fit models that I found. And the variables that went into the models were muscle score, size-corrected body mass, Julian date, the fat score, bird's age, time elapsed since sunrise, the year of study, the genera, the genus, which just means the migration strategy, and the bleed time. Here I present a graph of the analyses of covariance examining triglyceride concentrations for fat for each study year, 2013 and 2014, and the ENCOA graph for muscle score for both study years. And as reflected in the best fit models, for 2013, fat was a better predictor of triglyceride, and in the second year, muscle score was a better predictor for triglyceride concentration. In my opinion, one of the most interesting results of this study was the interannual differences in refueling rate. A bird's ability to refuel is dependent on a lot of different factors, but it includes the own bird's body condition. So let's examine this. In 2013, we saw higher refueling rates, but we also saw significantly later arrivals. We saw lower fat scores, significantly lower muscle scores, and significantly lower body mass. So birds were in poor condition in general when they were when they arrived on St. George Island in 2013. So this might indicate that they were compelled to try to refuel as much as possible, and it would reflect on the higher triglyceride concentrations that we saw. On the other hand, in 2014, we saw the lower refueling rates, and we also saw that birds arrived earlier, had higher fat scores, significantly higher muscle scores, and significantly higher size-corrected body mass. So in comparison, they were in a little bit better condition. So in this instance, St. George Island might have been functioning more as a refuge from weather or as a place to rest, compared to its function as a refueling site the previous year. I did not discuss this data, I'm not presenting this data here, but 2014 also represented the had higher overall species abundance. We had more species on St. George Island the second year. And this may have resulted in higher interest-specific competition, which may have affected refueling rate. On St. George Island, I found no age effects in arrival times, body condition, or refueling rates. And compared to the literature, this was somewhat different. The literature has found that there are age effects in migration times, departure from wintering grounds, arrival on the breeding grounds, the amount of time birds spend migrating and stopping over. And my data does not coincide with this. My data does, however, align more with this recent paper that came out of PNAS from Duffy et al. That found that a Gulf crossing is dependent on a lot of different factors, but not necessarily age. The effects of age on body condition, the literature says that this really depends on the species and season in question. And I did not find any age effects on any of these variables. So it really depends, as the literature says, on the time of the study. And finally, I found no age effects in refueling rates. And here, the literature consistently has found this to be the case. However, when I first started this, I expected to see a difference based on some foraging behavior of research by Heisey and Moore that found that gray catbirds, adults, are more efficient foragers than juveniles in the fall. And I expected that to reflect in refueling rate, but my data align with what other people have found. One of the other very interesting findings was the extent and consistency in the poor body condition upon arrival of long-distance migrants. And this is very similar to early stopover ecology work conducted in the Western Gulf Coast back in the early 90s. Moore et al. Found on Horn Island that Gutierrez thrushes and scarlet tanagers were the most commonly found dead on their carcass beach surveys. And as you now know, Gutierrez thrushes have really long-distance migrations, and so do scarlet tanagers. Coenzi et al. Worked out on East Ship Island, and she found that the keel of the sternums of many individuals, noticeably the rose-breasted grosbeaks and our friends the Gutierrez thrushes, protruded conspicuously. And that's a description of these photographs that we have from St. George Island that correspond with a muscle score of 0. And the fact that our birds did not, the body mass did not increase, or the refueling rate did not increase throughout the day on St. George Island, might be reflective of what some other people have found in other studies conducted along after ecological barriers, where lean mass reductions are not restricted to the pectoralis muscle, but also involved large reductions in the liver, the stomach, and the small intestine. And this is probably one of the more interesting things. It might reflect that these birds are physiologically limited in their ability to regain mass, because they're not going to be able to regain mass until they rebuild their digestive tracts. And this might be a very serious limitation in migration. Something that I found really interesting in the time that we spent on St. George Island was that in April 2014, the Florida Panhandle experienced record-breaking rainfall. It received 5 times the normal monthly rainfall in the period of 2 to 3 days. So let's look at the Panama City Station recorded 3.79 inches of precipitation in April 2013, compared to 13.95 inches the following year. And this is interesting in and of itself, but let's revisit the idea of the differences in age structure that I had. The first year we had a higher proportion of subadult birds. And the second year we had a higher proportion of adult birds. In 2014, the year that we had these crazy rainfalls. Whether these extraordinary conditions were only survived by the adult birds that were more experienced and resulted in increased mortality of young birds, or if the spectacular rainfalls created conditions where adult birds were obliged to stop, that's pure conjecture, but it is really an interesting thing to think about. It does, however, highlight the importance of long-term migration studies along the Gulf Coast, coupled with tracking environmental and weather data. All right, to summarize, we know that barrier islands of the Apalachicola Bay are indeed used by transgulf migratory birds in the spring. St. George Island serves as an important stopover site for migratory birds and may therefore be considered of important conservation, international conservation importance. We found that age-related effects did not drive the differences in arrival date, body condition, and refueling rate. Long-distance migratory thrushes consistently arrived in poorer condition than shorter distance gray catbirds, and that refueling rates were consistently similar between the two groups, but were different between years. To conclude, let's revisit the hypotheses of my study. I expected to see longer distance migrants arriving in poor body condition than shorter distance migrants, and the data did indeed support this hypothesis. I also expected to see higher refueling rates in lower distance migrants compared to the shorter distance migrants, and the data did not support this hypothesis. I expected to see sex and age-related differences, and for age, I thought that subadult birds would arrive later in the season in poorer condition and refuel at lower rates than adult birds, and the data did not support the hypotheses. And males would arrive earlier in poorer condition and refuel at higher rates than females, and I could not adequately test this hypothesis due to technical difficulties. Based on some of the results of my research, I would recommend we continue studies on St. George Island and other Apalachicola Bay barrier islands to better understand the dynamics of migrations at varying spatial and temporal scales. And these studies should consider interannual variations in weather, vegetation, and resource availability. Studies that do metabolite profiling should include multiple metabolites, including beta-hydroxybutyrate and uric acid, which are indicators of fasting and protein catabolism. And finally, I consider that the conservation, restoration, and management of our coastal resources should be considered an important aspect of the continental scale needs of these long distance migratory species. And last but not least, my heartfelt gratitude. My graduate work was made possible thanks to the funding and support of the NOAA Environmental Cooperative Science Center. I'm forever indebted to our on-the-ground partners at the Apalachicola National Estuarine Research Reserve, especially the incomparable Megan Lamb, who just went above and beyond to make our field seasons a lot more pleasant. I'm thankful for the support, encouragement, advice, and incredible patience of my graduate committee. You guys are great. The Heckscher Lab, Alan Neidel and Lori Lester were the best mates I could have asked for, were very helpful and supportive in the field. And finally, Drs. Seawagon, Moore, and Cohen, and Dr. Yeagle provided a lot of advice on and support on my field work. I'm very grateful to all of my supporters, and with that, I'll be happy to take questions or comments about my graduate work.
All right, good job. Any questions for Maria Mar?
How about these pictures, guys? Did you see the pictures?
Davina's got it.
Of course.
Oh, yeah.
So the males come in earlier. And this is kind of a standard thing for most migratory birds. And interestingly, in insects, a lot of times the males emerge before females. So what would you sort of hypothesize as an evolutionary reason behind that? Some adaptive reason why the males are getting
Yeah.
earlier to their
Yeah.
sites?
It pays to be early if you're a bird or if you're a male of your species, because the earlier you're there, the better chances you have of securing resources. And resources, I mean territory, defending territory. The earlier you are, you can find mates and you're not in a time crunch. And it's been proven time and time again in a number of species of migratory birds. And that, interestingly, it's not only about mating. It's also proven that in some species, the earlier you arrive on your wintering territories, the better you are. And this is especially true for birds that defend winter territories, like thrushes. Maybe not so much as warblers that have flocking behaviors in the winter, but it pays to be quick about it and minimize your migration time.
So if I'm a manager at NOAA or the near site in ecological, and I'm watching this, what would be, how could I relate this to management on St. George Island? I mean,
Yeah.
and part of that is clearly from your research, we know that things are going to vary from year to year. It's very unlikely that you only found the two, that we have a flat line and just in 2013 and 2014 things were different. It's probably, if you were to study this for 10 years, you'd get variation every year. So how could you take that and relate that to management or the importance of management at the near site, and not only there, but other coastal areas in the Gulf?
Well, one of the important things about St. George Island is that it's such a narrow island and land is at a premium. So having available resources for migratory birds is important. Considering that for their bird schedules, for protecting their land, for access to the public. The thing about fire escapes, which St. George Island is a fire escape, is this interannual difference that you see from year to year. And one year that might be very important for a migratory bird, the next year they may or may not use it. So not to consider that as, oh, well, the birds don't use it every year, so it's not important. It's actually very important, because when they do need it, it's fundamental. So this.
Good. So some of these, how far are some of these birds going? So they're coming across the Gulf. They're landing. Presumably the reason they land on these barrier islands is it's the first available landfall after a 1,000 or plus kilometer journey, and they're desperate to stop and feed. But where are they going afterwards? So
that's an excellent question.
So you did sort of allude to that, but maybe for the group here, how far are these birds going?
It depends on the species, and it depends on the individual, what population it comes from. But these birds are some of these birds are probably flying all the way to the northeast, or of course Canada, in the case of the thrushes. In the case of catbirds, they might just be going to North Carolina, or they might just be going to Delaware, because of the wider range of breeding distribution of these species. And then again, also the origin of the species might be very different. Some of these birds might be departing straight from northern Colombia or from the Yucatan, whereas some of the shorter distance migrants might be just coming from the Caribbean or Florida itself.
To add your question, so within the species, is there availability in how far they migrate? Because let's say you find these thrushes all the way from Delaware to way north, right, mid-Atlantic to the northeast. So is there any variability among the species? Look at it, as a species, they are not restricted to one geographical location, right?
Right. There's no way for me to know that from this data, but if we looked at stable isotope analysis, we could probably determine from the isoscape where the breeding destinations are. But the birds that are using this area, so during migration, these kind of stopover sites are like a bottleneck. So we have species from individuals from all over the population. So it's very hard to say that it's just one population, just the birds that breed in Delaware, or just the birds that breed in the Northeast border forests. It could be the entire continuum of the breeding habitat, or the breeding distribution.
So you suggested that the younger birds arriving later might have to do with like their aptitude, how savvy they are in getting across immigration. Has anybody discussed whether there might be different priorities, like if maybe not being in a rush is safer if you don't have many breeding prospects?
Yeah, that's a really good point. In birds, so the nomenclature is kind of confusing. We use juvenile, but they're really not juvenile birds. They are adult birds fully capable of breeding. So we call them second year or subadult birds. These birds, if they get to their breeding and establish a territory and find a mate, they're very much capable of breeding. So it is in their interest to breed as well, their first spring that they're able to. But I know that that's a question that a lot of these people that do the geolocator stuff, like that wood thrush study that I presented in the introduction, that's one of the things that they look at, is the whole carryover effect and the carryover implications of how fast you winter, where you winter, how fast you migrate when you arrive to your breeding destination. And it's all about migratory connectivity, and that's where it's at.
And presumably the younger birds are less experienced. I mean, that's kind of what we assume.
Right. And that's really the case in the fall.
So the mortality is then maybe higher.
Yeah, the mortality is higher, and it's really obvious in the fall, because in the fall the birds are, right out of the nest. It's the first migration. A lot of them die, but in the spring they've already survived their wintering, overwintering time. But they're still less experienced. Still their first migration north, so there is still a lot of mortality for young birds. Yeah.
Which you think would be the main, the more important effect of sea level rise in Sandra Island to the birds? Like, which you think will be the first thing that they will be lacking?
Habitat.
In
Habitat, absolutely. That's going to be the main problem, the main issue is the habitat on the island already is really restricted for what they can use. And if we have sea level rise, we're not only going to lose mass of the land, it's going to change the vegetation structure because of saltwater intrusions and things like that. So it's going to have a detrimental effect and loss of habitat for these birds. And in the pictures where you see the pectoralis muscle completely depleted, that might mean mortality for that bird, that extra little bit that they have to go, the extra 4 miles to the mainland. And then presumably with sea level rise, the mainland will be even further than that. So it might really spell disaster for some of these individuals.
They will not have any options in case that the reduction of the island comes to a limit, that they will not have any option. It is completely disappear or is very reduced. Like
Yeah, there won't be an option. There will be a high pressure for birds that can make it all the way. There will be selective pressure for birds too. And so this is that aspect is really interesting because the amount of fat that a bird deposits before crossing the Gulf is important for them to make it all the way. But if we're seeing habitat reductions, not only because of sea level rise, but because of human development and all of these things, we see it in the breeding grounds, in the wintering grounds, in the migration routes. So the quality of habitat in the wintering grounds, when they're depositing all of these fats to make their migrations, is also important. We're having all of these effects all over the place. So it could spell big problems.
I was just wondering how we didn't choose a second site. You did St. George Island, right? There's a lot more sites in the Gulf of Mexico, further. There's Florida, in Mississippi, Alabama, Texas, And they're all part of ECSC and near sites. So I was just wondering, and they give access to boats and people who work there. I was just wondering why they didn't choose a second site.
Well, for
For
doing this
comparison
it's logistically difficult because the number one priority is the safety of the animals. That's just when you're working with animals, especially with birds, your eye clock would be full if you just have a bunch of birds dying. So it's very important to be safe about this. And to run it safely, you have to have a well-trained team. And I was very lucky to have an awesome lab that had really well-trained people taking blood samples, checking nets, making sure that the birds were managed safely and released really quickly so that they can continue on their way. So the logistical problem is probably the big answer to that. The other part of the equation is that there's a lot of stopover ecology work along the Gulf Coast. The labs out of Mississippi, Dr. Moore, just a crazy amount. They're so prolific. They have a lot of stuff out there. But the majority of the work is concentrated on the west part of the Gulf Coast. And for many years, it's been thought that, oh, the east part of the coast is not as important for migratory birds. So our lab decided to, well, let's see if that's true, because it doesn't seem like it would be. And from the data that we're coming out, this paper by Dr. Lester, published in PLOS ONE as part of it, this work, Alan's work, and Dr. Hector's new grad students all point to the direction that no, the eastern Gulf Coast is important for migratory birds as well. And it would be very interesting to have these comparisons throughout other ECSC sites for sure. I know that the new grad students are looking at St. Vincent Island, which is within the Apalachicola Bay Area, to compare these stopover sites. But for a first step, we just looked at St. George.
So one of my goals is to bring awareness to this, because you have thousands, if not potentially tens of thousands, if not more, birds that are in these on these islands, and you can't see them because they're secretive. The vegetation's really dense. If these were shorebirds out in the open, we would have known decades ago about how important Apalachicola Bay is. But we have to set up nets in dense vegetation, leave them alone for 20 minutes, and wait for the birds to fly in. And you can be amazed how many birds you can get in a net. You had no idea they were even there. So this is like a hidden resource that, one of my personal goals has been to show that these eastern coastal areas are just as important, perhaps, as some of the western Gulf Coast stopover areas.
Question about that. What would be the possibility of, you're saying they're secretive, but they vocalize at some point, right? Or not?
They don't do a lot of vocalizing. Sometimes they do.
Okay.
But you can
So
walk
an
down a
acoustic
trail and
survey might work, might not work.
probably not very well.
Okay.
Maybe some species. But we can set up a net and not know there's any bird around. You come back 10 minutes later and you have 4 birds hanging in the net.
Right.
As soon as you leave, they start moving around again.
But that rain is really important, actually. Like you said, you had a big rainfall in 2014. That's actually really important to Apalachicola Bay. But it's actually further up north, because of the Apalachicola River.
Mm-hmm.
Because you can have fresh water all the way out to the Gulf of Mexico because of that river. So.
Any other questions for Remar?
I'm curious how many Swainson's thrushes you included in your first year, because I guess some of them winter in Central America, so they're not as long distance migrant as the other Cathar's thrushes. So couldn't conceivably some of them be migrating less than some of the catbirds?
Yeah, it's very true. The Swainson's thrush of the Cathar's thrushes, they have would have a shorter distance, because some of them they do winter in Central America, but they also go somewhat into Colombia. There's no way for me to know which one's is which at the point. I don't know the number off the top of my head, but in the first year, the bulk of the data was from Swainson's thrush, because they were the more abundant thrush that I had that year. So it was just a matter of what was available. But that's an interesting point, because there was no difference in any of the variables that I looked at between the Swainsons, the Beery, and the Great Sheep. So they were all similar in all of their other body condition variables and body size variables.
If you could have chosen one more species to look at, what would you have chosen? Beyond those four?
That's a really tricky question. Now looking back, I would have picked the yellow-billed cuckoo and the northern water thrush. The yellow-billed cuckoo because it's really cool, and the northern water thrush because now, in retrospect, I can see we have a large sample size. But that's the problem with migration, is that you can't predict the numbers that you're going to have.
Do you think that the island is just as important for those species, or
Yes,
just the thrushes?
absolutely.
Okay.
No, I think that the thrushes are a good representation of what happens to the long distance migrants. So rose-breasted grosbeak, the tanagers, the scarlet tanager, the yellow-billed cuckoo is not a long, yes, all of these species. I think that what's happening with the thrushes is a good indicator of what's happening with the other ones.
And how many migrant species did you end up with?
Oh,
Did our lab end up with during your?
I can pull that off from my data. I cannot remember off the top of my head those numbers.
Okay.
I think
Alan, do you remember how many species we've had total out there?
2013, we had 38 different long distance species, and in 2014, 41.
Okay.
So I think cumulatively probably 45 or so.
And that's comparable to other places on the
Yeah, it is.
on the coast.
Mhm. Yes. Melanie, you had a question.
Yes, so you said that in 2014, you had the regression of the size-corrected body mass with a date, and you said that lighter birds arrived earlier. And you did not have that same pattern in 2013. But weren't the banding dates a little bit later in 2013?
Yeah, they were, absolutely.
So do you think you maybe, it's possible that you just missed the lighter birds?
Yes, that's exactly. I didn't want to go into it during the talk because it's a cool little data point, but in my final document, that's definitely something that was driving some of these differences. We, so for logistical aspects, the lab were students, so we have to have classes here and take care of that kind of stuff. And it coincides with migration. So the first year we were there for 3 weeks, and then the second year we were there for a little bit longer and a little bit earlier. And that's where we could probably see those differences. But that's in the catbird. And the thrushes, the time that we were capturing them, it overlapped between the 2 years. So that was not what was driving the thrush data, but it definitely is possible that that was what happened with the catbird data.
Any more questions for Remar? All right, so the committee's going to meet, I guess, in this room with Remar for a while, but just give her a hand. Thanks, Remar.